Hello, this is Mr. Eschholz again, and welcome back as we continue our AP Bio. And we just finished up a section on kind of some chemistry review, the chemistry of water, pH and stuff. And now we're going to get right into organic chemistry, and we're going to talk a little bit about carbon chemistry and why it's so important to the biomolecules, because that's where we'll be heading next. So if you look at this diagram here, it's a good cartoon. There's a good series on NPR about carbon in general and how it bonds and stuff that you should take a look at. But the big thing here is it's got four hands. It's got four bonds that it could potentially make with other molecules or other atoms. So if we look at it, carbon is the basis of organic compounds, things that contain carbon. Originally, they thought it came from only living things. We know now that we can generate them synthetically, but we'll go with that definition for now. So carbon is so versatile as a building block because it has a valence of 4. It is what's said to be tetravalent because it can make 4 different bonds. It has 4 unpaired electrons in its valence shell. So it can build a bond branch in 4 different directions. It forms the backbone of organic molecules. The carbon-carbon bonds can form chains that can be straight, branched, or rings. And depending upon the shape, it's going to affect the function and how they interact, whether or not it's in an aqueous solution or whether it's dry. And so if we look at this next picture here, we can see some of the examples of, here's some examples of just hydrocarbons. So you can look through, it's just carbon and hydrogen. So we see we've got a number of them that are straight chains. So those carbon are straight. Here's one right here that is branched. And then we've got a couple, the cyclohexane and the benzene, that are circular. So carbon, because of those 4 bonds, can take any number of different shapes that affects the function and how it's used in biological systems. And here's the Lewis dot structure for carbon and why it's so important. So again, we mentioned this word tetravalent. Whoops, let's see if I pen, there we go. Tetravalent. It has 4 valence electrons. And so it can form 4 different bonds. So each of those unpaired electrons represented by the dots can form a bond in 4 different directions. And if we look at some of the other compounds that, elements that are necessary for life, basically carbon, oxygen, hydrogen, and nitrogen, we can see that the valence, the number here, we talked about carbon being 4, so it can form 4 bonds. Nitrogen has a valence of 3. It has 3 unpaired valence electrons, so it can form 3 bonds. Oxygen has 2 unpaired valence electrons, so it can form 2. And hydrogen, with its 1 valence electron, can form 1 bond. So those are some important numbers to keep in mind as we go through and start to put these atoms together in different combinations to make the macromolecules. So here's an example, top's a little bit fuzzy, but here's the Lewis dot structure for carbon dioxide, CO2. So there's carbon in the middle with its 4 valence electrons shown in black, and then there's 2 oxygens on either side with its 6 valence electrons shown in red. And because there are 2 unpaired, that's how many bonds that oxygen would like to form. So if we match up, if we form covalent bonds and share, we could form 1 pair right here. We could form a second pair right there that'll form a double bond to the left of the carbon, and then the same thing will happen on the right side. There's 1, there's a second, the double bond right there. So if we look in the middle, it's showing the same thing, but what it's showing is it's showing those 4 electrons in that double bond being shared right here, and these 4 right there. And if we go down in the bottom and look right here, we're seeing the same thing, and here's our double bond on either side. And then there's our lone pair, so there's the Lewis dot structure of carbon dioxide. But again, the number of valence electrons is key for looking at carbon chemistry. So hydrocarbons is one category. When we talk about lipids, it's almost entirely hydrocarbons. There's very little oxygen. So they have only hydrogen and carbon. So they're hydrophobic, they're non-polar, they don't like water. So we're talking about petroleum and lipids. And here's a couple examples of very easy ones. So here is methane, the simplest of the hydrocarbons, and you can see we've got carbon with its 4 valence electrons in blue. Each hydrogen has 1 valence electron shown in red, and together they share so that carbon ends up with an octet. It becomes stable by sharing 4 electrons with hydrogen. And here is ethane. And it's the same thing, except now we've got the 2 carbons in the middle, and it's sharing with 6 hydrogens to fill its octet. So here is kind of different ways of showing those hydrocarbons. So the ball and stick model, the structural formula, the space filling model. I'm not going to say anything else about this. You might want to pause it real quickly and just look at these and just understand there's different ways of representing carbon and the structures in macromolecules. So the properties of carbon compounds and macromolecules are determined by 2 main categories. The first is the arrangements of the carbon skeleton. So what's the type of backbone? What's the structural arrangement? So we said before, we can have linear, we can have branched, and I don't have a picture right here, but we can also have cyclic or circular ones that will go like benzene. Where we'll go, and we could have double bonds in there as well. So the structural arrangement is huge for carbon compounds. The second one is the pattern. So we can have compounds, if you look up here with letter A, these are called structural isomers. And if you count the number of carbons, you can see there's 4 in each. If you count the number of hydrogens, we've got 8, 9, 10, 3, 6, 10. H10, they both have the same structural formula, but they're put together differently. The pattern is different. These are what are known as structural isomers. In the middle, they've got the same molecular formula, but now they're rearranged slightly different around a central double bond. So the only thing here is that they're flipped top and bottom, and I'll show you a second picture in just a second. And those are called geometric isomers. And the last category are kind of the toughest, but they're the most important biologically speaking. So here are structural, they have the same structural formula, but they're said to be enantiomers. They are mirror images of each other. They can't be superimposed on each other, but they can kind of fold over as in mirror images. And when we talk about some of the biologically active macromolecules, this is going to become very important. So here's an example of structural isomers. All 3 of these compounds have the formula C5H12. And you may want to pause the video at this point and just count them up to double check. But we have linear, branched, doubly branched, and so same formula, different structure. Here's an example of C6H12O6. So you've all have probably heard of glucose. All of these different sugars up here have the same formula, C6H12O6, but they're all put together slightly differently. So fructose you probably have heard of, but there's other sugars like galactose, allose, altrose that have the same formula. They're just put together slightly differently, so they're structural isomers. Here's what I talked about the geometric isomers. So here's our double bond. And if we look at the top one right here, we've got the 2 hydrogens are both facing down. That's what's called the cis position. And if we were to break that double bond and flip this side of the molecule, now all of a sudden we've got the H up. Those are geometric isomers. They're put together similar, but they're rotated around a double bond. So the cis or bent form is right here, what we see like that. This one shows it going up. And then there's the trans form. And the way I learned them was the boat and the chair. And when we get to lipids, especially, we're going to talk about trans fats. So you've probably all heard about that. We're going to actually show you what does something mean when we say it's a trans fat. And then finally, here's the idea of these enantiomers. Here's your mirror images of each other. And the key for that is there's a central molecule of carbon, and there are 4 different things bonded to it. And they can't be superimposed, but we can get a mirror image of it. And in class, we'll play with models a little bit, so hopefully that'll make more sense. Here's another picture. If you look at your hands, it's hard to place one hand on top, back of hand to palm, but if we go palm to palm, the thumbs will match up, and we can get mirror images of each other. And that's going to become important because a lot, as I said before, a lot of the biological molecules. So here is L and D dopa. And DOPA is a drug that's effective against Parkinson's disease. And only the left hand, the L version, is going to be active. The D version, the right-handed version, is not going to be active. So you could give this one here as a drug, but it's not going to be effective against Parkinson's. Only the L version, the left-handed version, is. And here's just a quick kind of breakdown of isomerism. And again, you can see the structural isomers over on the left, the stereoisomers, that they've got the same structure and bond sequence, but there's rotation around the double bond for geometric or optical, the enantiomers. So you can pause the video, look at that for a second if you want. And getting near the end here, so one of the last things that we're going to look at, so far we've kind of talked about how the carbon's put together. I liken that to the Christmas tree. We've got the structure of it. Is it a branch? Is it a straight chain? Is it circular? Now what we can do is let's decorate the trees. And the way we decorate the trees is there are these things called functional groups. And the functional groups are kind of added onto the side of the carbon chain, and they give it certain properties. The most simple one, and one that we'll spend a lot of time on, is the hydroxyl group, OH. And this makes something an alcohol. And it also makes it water soluble, which is going to be very important. So if you see OH groups like we do over here on the right, that's going to be water soluble, and that's usually found in carbohydrates. The second group is the carbonyl group, and this is found in lipids and carbs, and it's carbon double bonded to an oxygen. This one's a little bit trickier because there's two possible categories. There's something called an aldehyde or an aldose, because sugars end in ose, and this is when the carbonyl group is at the end. So there's your C double bonded to O, and there's always a hydrogen with it. The other case is you can have a ketone or a ketose if it's a sugar, and here the carbonyl group is in the middle, and there's no single hydrogen attached to that. So the aldehydes, you may have heard of formaldehyde, and the ketones, you may have heard of acetone, are two examples that you may have seen, but there's a carbonyl group. The next group is the carboxyl group, and this is found in proteins. This makes organic acids. So we're talking about amino acids there because it is organic. And here is the C double bonded O, OH, and many times this will be abbreviated as COOH, like that. The next group that you need to be able to recognize is the amine or the amino group, and this one is pretty easy to recognize because you got the nitrogen in there. And here's where we have proteins. So the amino acids have the carboxyl group that we talked about before, but they also have an amine group, and that's what makes them amino acids. And the last functional group that's going to be very important is the phosphate group, or PO4. Okay, and this one is we're going to find in nucleotides, DNA, ATP, a lot of the molecules of heredity and energy we're going to talk about. So these are the 5 functional groups that you need to be responsible for, and make sure that you can recognize and understand what properties they give to different carbon molecules. And the last slide that I'm going to show you is we talked about the organic acid. So here's lactic acid, which you probably all have either felt or heard of before. And so here's your C double bond OH. And what happens is in the body, this hydrogen, remember we talked about, what did we say acids were? Acids are proton donors. Well, if this acid gives up that proton, it becomes ionized. So over here it becomes O minus, and there's why it's an acid, and this becomes lactate. And so in your body, we talk about how it's lactic acid, and really it's actually lactate because it becomes ionized, and that's the burn we feel in our muscles as it becomes ionized. The same thing is going to be true. We're going to talk about the citric acid cycle. Well, the citric acid will break down into something called citrate. We can talk about acetic acid, which is vinegar. So here's the formula, CH3COOH, and what happens is this will dissociate. It'll give up that hydrogen and become the acetate ion. So as we go through, just keep some of these things in mind in terms of the carbon chemistry, and that's where we'll leave. And I will leave you with the formula or the table right here for that. Thanks again for your time.